Welcome to this Fast Facts presentation from Cell and Gene Therapy Insights and Lonza. I'm Charlotte Barker, an editor at BioInsights, and joining me today are Andrea Toll and Timo Glasner, who are here to talk about the 4D Nucleofector LV unit, efficient, non-viral, large-scale transfection for cell and gene therapy applications. Andrea Toll is a senior product manager at Lonza Cologne and is responsible for Lonza's Nucleofector transfection portfolio. In her current role, she's focused on large volume transfection and cell and gene therapy applications such as CAR T cells, genome editing, and GMP manufacturing. Timo Glasner is Associate Director, Engineering within Bioscience Solutions at Lonza Cologne and responsible for the technical development of Lonza's Nucleofector transfection platforms. In his current role, Timo leads the development of the 4D Nucleofector LV unit for large-scale transfection and has technical responsibility for the product design of the 21 CFR Part 11 compliant software, as well as the therapeutic consumables for GMP manufacturing. If you'd like to ask the presenters a question, simply use the Ask a Question box to the right of viewing pane. And with that, I'll hand over to Andrea and Timo to introduce themselves and kick off today's presentation.
Hi, my name is Andrea Toll, and I am one of the global product managers for the Nucleofector technology with focus on cell and gene therapy. Today, I have my colleague Timo here with me, and together we are showing you the 4D Nucleofector LV unit to demonstrate you how it works.
Yeah, hi, my name is Timo Glasner, and I am Associate Director Engineering within Bioscience Solution here at our Lonza Cologne facility. And today I will show you some videos demonstrating the use of the device and its consumables.
Okay, before starting with the hands-on part, let me provide you with some background on the Nucleofector technology and especially the 4D Nucleofector LV unit and how it can support cell and gene therapy applications. Our state-of-the-art Nucleofector technology, it's a non-viral technology which enables highly efficient transfection of primary cells combined with high cell viability. It has been cited in over 10,000 publications, many of them high-ranking, and is used in renowned award-winning labs working in the cell and gene therapy space, for example, Yamanaka Lab with iPSC generation and Charpentier or Doudna for CRISPR. The technology can be easily adapted to different substrate types, be it plasmid, DNA, mRNA, or ribonucleoproteins. And since it allows for efficient co-transfection of these substrates, it's the ideal platform for genome editing using transposons, zinc fingers, talons, or CRISPR. The platform supports single reaction formats to high throughput formats for e.g. CRISPR screening. And also scalable volumes per reaction from as low as 10,000 cells up to 1 billion cells. The platform always consists of a Nucleofector device, a cell type specific Nucleofector solution, and the respective nucleofection vessel, plates, strips, cuvettes, or cartridges. In the cell and gene therapy environment, the technology has been widely used for basic research, but now has also evolved into manufacturing processes exploring non-viral ex-vivo modifications, since it's easily scalable to higher cell numbers in the range which is of interest for autologous cell therapies. The application can be established in smaller scale formats, as you can see here on the left-hand side, to save cells and substrate, and then scaled up to the larger scale formats serving 100 million up to 1 billion cells. Our large-scale platform, the LV unit, comprises the core unit, which is the controlling unit, and attached to it, it's a large volume functional unit called LV unit. And here you can already see the next generation for the Nucleofector system, but the setup and procedures shown by Timo in a minute do not differ from the first generation, except that the graphical user interface is a little more convenient to use. The LV unit can handle two different nucleofection vessels. The 1 milliliter cartridge you see on the left-hand side is suited for transfecting a fixed volume of 1 milliliter. It can be used for up to 200 million cells. And the LV cartridge that you see on the right-hand side is the flow-through version, which can transfect up to 1 billion cells in volumes of up to 20 milliliter. And transfections are done in successive fractions of 1 milliliter. So now, Timo, would you please take over and demonstrate the device and how it works?
Sure. So I have been to the lab and taken some videos of the 4D Nucleofector LV unit. Let me just walk you through them one after the other. So let's start with the 1 ml formed. The 1 ml cartridge is filled manually via a sterile injection port. It is filled from bottom to top in a semicircular reaction room. The filter is there for venting. Now I will show you how to process a sample with the 1 ml cartridge. For loading the sample, hold the cartridge in an about 45-degree angle with the port at the bottom. Carefully insert the pipette tip into the port in a 90-degree angle and slightly circle the pipette to simplify the piercing. Slowly fill the cartridge with 1 ml sample. Stop once you've observed liquid above the reaction chamber. Insert the cartridge into the slot. Ideally, push it in the middle of the handle to make sure that it is inserted straight and both levers, the upper and the lower, audibly snap in to allow for proper contacting. Press start to initiate the nucleofaction process. Once the nucleofaction process is finished, remove the cartridge by pushing both levers. Hold the cartridge in the same way as before when loading the sample. Insert a fresh pipette tip and remove the sample. We are now getting to the flow-through format. The LV cartridge uses the same reaction chamber as the 1 ml cartridge, but is equipped with tubing for automated loading and clearing. It can process a sample size between 3 and 20 milliliters. The sample is processed in fractions of 1 ml. When using 20 ml, up to 1 or 2 billion cells can be transfected. It has 2 input lines. One line is for cell suspension, and the second line is optional. We recommend it using in case you work with mRNA, as it keeps mRNA and cell suspension separate until shortly before the transfection step, and thus avoids degradation. Let me briefly show you our dedicated LV reservoirs for sample loading and collections. For sample loading and collections, we recommend the use of our LV reservoirs. Alternatively, bags may be used, but especially on the input side, the reservoirs have several advantages. You can stir the sample to keep an even cell suspension and avoid sedimentation. You can fully clear the liquid and reduce sample loss. The Therapeak version of it is fully closed and is principally like a solid bag. Now I'm going to show you how to prepare the LV cartridge for sample processing. For mounting the LV cartridge at the LV unit, first insert the cartridge into the slot in the same way as shown before for the 1 ml cartridge. Make sure that upper and lower snap mechanism audibly engage for allow proper contacting. Attach the venting tube with the filter to the male lure connector on the front plate. Insert the tubes into the 3 pinch valves. Ensure they are inserted completely. If you started defining your experiment setup already, the software can support you in inserting the tubes into the pinch valves by automatic opening the valves. Alternatively, you can open the pinch valves manually by pushing the black button on the center. Insert the T-connector into the T-connector holder. Insert the larger 2 diameter tubes into the black holders and ensure the tubes are inserted completely into these black holders, as the lower holder comprises a light sensor for liquid detection in the lower tubing line. Insert the upper tube into the upper pump and close the flap. When closing the flap, gently hold the tube without pulling it. In case you use the second input line, repeat the step with the lower tube. You may also establish the connection of the LV cartridge to the input and output reservoirs prior to cartridge mounting at the device. In that case, please make sure that the tubes are clamped to avoid liquid movement. The next step is establishing the connections to the LV reservoirs. The LV reservoirs can be connected with the LV cartridge either manually or via sterile welding, which is not shown here. Make sure that all clamps are closed. For manual connection, remove the cap from the spinning spirals of the cartridge and connect it to the swappable injection port of the reservoir tubing. The twist opens the connection in a sterile fashion. Do so for all connections. You typically start with the collection reservoir pre-filled with medium. The input reservoir, holding the cell suspension, comes last, shortly before starting the nucleofection process. As a last thing, I will now show you how a sample is processed using colored liquids. Red is representing the cell suspension, blue is representing the substrate. So once all connections are established, open the clamps and start the nucleofection process. After calibration, the sample gets pumped into the cartridge based on volumes and volume ratio entered into the user interface. Via the upper pump, the system first starts pulling liquid from the cell suspension input reservoir. It stops once reaching the cartridge and gets retrieved back to the T connector to wait for the substrate. Via the lower pump, the system now starts pulling liquid from the substrate input reservoir. Once it reaches the T connector, both cells and substrate are pumped jointly into the cartridge based on their volume ratio. The first 1 ml fraction then gets transfected. Afterwards, the transfected fraction is pushed out of the cartridge via air, going through a sterile filter into the collection reservoir that has been pre-filled with culture medium. And then the next 1 ml is filled, and so on, until the whole sample is processed. And that's basically it. You've seen the 4D Nucleofector LV unit live in action. And with that, I hand it over back to you, Andrea.
Thanks, Timo. I would like to close our hands-on session by summarizing some major benefits of the technology for setting up cell and gene therapies. The Nucleofector technology and the LV unit supports 5 key success factors. The technology achieves robust and high transfection efficiencies in cell types relevant for cell and gene therapy. It is versatile as it has been proven for use with DNA, mRNA, protein, and thus supports applications like CRISPR and CAR T-cell generation. And it is suited for many primary blood and somatic cells. With its different platforms, it's scalable, which means you can smoothly transition from research scale to manufacturing scale by using the same conditions in any scale. And it is safe because it's a non-viral genetic modifications and consumables are fit for purpose. That means available for research and GMP use. Last but not least, we offer documentation and support. We can provide technical and application support by our highly skilled scientific support team. We are offering IQ/OQ services for the device. The system can be operated under a 21 CFR Part 11 compliant software. And we offer regulatory support packages with more detailed product information, and a master file is in preparation for next year. In addition to the Nucleofector technology for the genetic modification step, Lonza offers further products and services that can support many of your workflow steps during process development or manufacturing. We have highly characterized primary immune and adult stem cells, either as immediately available cryopreserved cells or as made-to-order products to set up your workflow. Our serum-free therapeutic media and reagents enable robust, consistent, and cost-effective cell culture. The Cocoon platform is a closed and automated platform for cell expansion that can provide substantial cost reduction in manufacturing cell therapies through a functionally closed and automated platform. We are currently working on linking this to the LV unit. For the endotoxin testing, We offer regulatory compliant bacterial endotoxin testing products and services to ensure your cell therapy can be used with confidence. With the cost-effective and easy-to-implement MODA solutions, paperless, GMP-compliant data capture in laboratory and manufacturing can be fulfilled. Last but not least, we offer extensive cell and gene therapy services from process development to GMP manufacturing. Thank you all for joining our demonstration of the 4D Nucleofector LV unit. If you have any questions, please post them on the right-hand side of this video or submit them to the link that is shown here, lonza.com/transfection scaleup. Thank you again and bye.
Thanks. Bye.
Thank you, Timo and Andrea, for a great presentation, and thank you for watching today. If you'd like to download a poster version of today's talk summarising the main points, you can find that below the video. We hope you'll join us again soon for more Fast Facts.